Welcome everybody, I'm Brian McCourt, President of the McCourt Foundation. And on behalf of the entire foundation, we'd like to welcome you to the 2022 Educational Update. Today, we will have some of the finest minds from Mass General, Brigham and Women's, and Cedars-Sinai presenting on some of the latest topics in MS, Alzheimer's, and ALS. I'd like to take an opportunity to thank all of our sponsors for being a part of this day, but especially our major sponsors, that being UBS Financial, EMD Serono, and MT Pharma. But again, all of the corporations that are being a part of this and making this day so successful. As you all know, our mission is to help cure neurologic diseases. And this is why events like this are so important to us. Today, we'll be broadcasting a virtual portion of this day, so it gives us the ability to have an in-person and virtual option. So we'll have people from all over the country being a part of this. It's exciting because, again, it's important to get the latest information out there. So thank you so much to all of you for being a part of it. Get ready for some exciting information. Take advantage of all the Q&A. And thanks again for being a part of the McCourt Foundation.
Thank you for joining us today at the McCourt Educational Forum. We really appreciate you coming out. My name, as Kelsey said, my name is Seif. I'm Senior Vice President at UBS Financial Services. And we're excited to be here to help sponsor this educational update. We believe that the financial service industry can not only help families plan for the financial future, but we certainly like to support and help advance the critical research and awareness of these various neurological diseases we're hearing more about today. You'll be hearing from some of the top doctors from Mass General Hospital, Cedars-Sinai, and Brigham and Women's Hospital on the progress and developments of their ALS, Alzheimer's, and multiple sclerosis research. I personally am impacted by neurological diseases of family members that have MS and Alzheimer's. So I'm thrilled to be here to help sponsor this. At this time, it gives me great pleasure to introduce Dr. James Berry, the Chief of the Division of
So I think we've got a really exciting panel of speakers today. We're very excited to share some of the really most cutting edge research that's going on in ALS today and put that in the context of how we're developing drugs to try to combat the disease and come up with therapies. There are going to be three of us speaking, Clotilde Lagier-Tourenne first, then I'll speak, and then Sabrina Paganoni will speak at the end, and then we'll have a question and answer session that we're excited about. I'm going to introduce our first speaker, Clotilde Lagier-Tourenne, who is an associate professor at Harvard Medical School, and we'll be talking about mouse models in ALS and how they guide us to new therapies.
Thank you very much, everyone. It's really a pleasure to be here today, and I'm grateful for the invitation from the McCourt Foundation to discuss advances in ALS. And as Dr. Beria said, I'm going to discuss mostly how important it is to understand disease mechanisms and to model disease to develop therapeutics. While Dr. Berry and Dr. Paganoni after me will discuss advances in clinical development. So, amyotrophic lateral sclerosis is also known as Lou Gehrig's disease, in particular in US, from the famous baseball player who has really raised awareness on this condition. In France, we actually call it Charcot disease from the first neurologist who described the disease more than a century ago. This disease is characterized by progressive paralysis. It's a very severe condition that leads usually to a fatal paralysis within a few years, but there is a large heterogeneity in the patients. And while most cases start at adult age, there are some very severe cases also in teenagers. it's really due to the death of specific neurons that we call motor neurons. And so they are these neurons that go from the brain to the spinal cord and from the spinal cord to the muscles, and they are necessary for muscle contraction and for movement. So what's very interesting is that also we have very well clinically defined syndrome. In the last decade or so, it was really obvious to the field that actually ALS shares a lot of common features with another neurodegenerative disease that is called frontotemporal dementia. And this is a very different disease where the neurons affected are not the same. The patients do not present the same. They have a language and behavioral dysfunction. But there are many insights that tells us it's actually a common disease with many different aspects that are shared. And indeed, for many years, we knew that some patients can have both ALS and frontotemporal dementia, either at the same time or later, a few years later. And also some very interesting families were studied where some patients had either ALS while other patients had frontotemporal dementia. And it's really the advances in genetics in the last 20 years with the discovery of many genes responsible for ALS and frontotemporal dementia that have shown that actually the same genes can lead to either of the disease. And really the field that we don't understand yet really what, for us in the same family, some patients are going to develop either disease, either ALS or frontotemporal dementia. These genetic studies were really fundamental for us to understand what happens, what are the causes that really lead to the death of these neurons. And you can see there are several colors here that really just tells us that these genes, what they point us towards some mechanisms that are very important for the cells. And this allows us to study why, when these mechanisms are altered, the motor neurons die. And that's a genetic study, it's really fundamental. But there are also a lot of insights that have actually been obtained by studying post-mortem tissues from patients. And so what we knew for a very long time is that when we look at the neurons in these patients, either with ALS or with frontotemporal dementia, there are some abnormal aggregates that can be stained. For a very long time, we didn't know what were these aggregates. And the breakthrough really came in 2006 from studies at UPenn and in Japan that identified that these aggregates were mostly formed by a protein that is called TDP43. The reason I'm saying it's really a breakthrough is what you can see here. Basically, almost all patients with ALS have these abnormal aggregates of TDP43. And a large proportion also of frontotemporal dementia patients also have, which was an additional argument to say, these two conditions, they have some common features. So this also observation in patients was very interesting because normally TDP43 is in the center of the cell in a compartment that we call the nucleus. But you can see here that it's in brown. It does go somewhere it should not go in the cell. So it's accumulating at the periphery of the cell. And what this observation already told researchers was what's happening when TDP43 does that? What is the normal function of TDP43 in the nucleus? Why, when it goes there, it does kill the neurons? And that are questions that we cannot answer in patients. And this is really where modeling the disease becomes very important to answer this type of questions, as well as to understand what these different mutations that I've shown you in many genes lead and why they kill neurons. And so we have different ways of modeling disease. Some, for example, just in a tube in vitro, some in cultured cells, including cells that we can culture from patients after taking some blood or some skin biopsies, but also some more complex organisms and animals that allow us to assess really what's happening during disease. And really this modeling is very important, as I say, to understand what are the events that leads to the motor neuron death. It does also allow us to understand why there are different mutations and why there are different ways of killing a motor neurons. Basically, the ALS is seen in patients when the motor neurons die, but you can imagine that there are many ways of killing a motor neurons. And so these models allow us to really understand what are the different ways, because the therapy is going to be very different depending on what is the cause. And we need to understand these different subtypes to develop really efficient therapies. We also need to understand what happens, because we want to know what our drug is supposed to do if we have no idea what we want to change in the cell, right? And so that's what we call therapeutic targets. You may read a lot about that, identifying new therapeutic targets. It's really what it says. It's like identifying what we want our drug to be doing in the cell to prevent that the cell die. And what I will show you now in the next few minutes is several examples where these models have been used to really lead to therapeutic development. And how we use now, including mouse models and other animal models, to also inform how the clinical trial, clinical development is being done in patients, and how we can really identify drugs and move them from the laboratories to the patients. So this is an example, and I think a very nice story that started from researchers at Stanford and at UPenn, who were really wondering what is actually happening when this protein I've mentioned, TDP43, that is abnormal in all the patients, why is it killing the cells? And so indeed, they used first yeast. And so they showed that if you express TDP43 in yeast, the yeast cannot divide, they don't form colonies. And an advantage of the yeast is that you can very relatively easily either inactivate or activate any genes you want. And so they went for a fishing expedition. They had no idea what would help with TDP43 toxicity. So they inactivated each gene in yeast, and they looked at which one are going to help the yeast to survive when there is TDP43. And they came up on a gene that indeed when it's inactivated, the yeast, despite TDP43, are going to live. And this gene is called ataxin-2. Then they actually used Drosophila, where the researchers really can look at the eye, and when TDP43 is expressed, the eye degenerate. And again, if we remove ataxin-2, the eye is rescued, the eye phenotype is rescued, despite the presence of TDP43. So you would think, oh, that's really far away from human disease, right? Yeast, Drosophila, why do we care? Actually, the researchers went back to ALS patients, and what they realized is that when you look at the gene encoding for ataxin-2, there are some changes that lead to an increased risk of ALS in patients. And so they really linked this ataxin-2 function to ALS. And it was very interesting because actually ataxin-2 was not known at all to be involved in ALS, but was previously described in another neurodegenerative disease with gait abnormalities. And very importantly, just by this fishing expedition, first in yeast, now we know that we want to remove ataxin-2 to lower ataxin-2 in patients. And so they have actually developed a method that is based on antisense oligonucleotides. They have tested that in mouse models. And now there is a clinical trial that is led by Biogen in patients with ALS. And I told you, TDP43 is abnormal in almost all patients. So really this is an approach that may be beneficial in the large majority of patients. So I've shown you this story from the beginning to the clinical trials that was starting in yeast. There are many other examples. Of course, today I cannot go through that, but we are also using similar approaches in worms. You can see that worms that express mutations, that are the mutation we see in some ALS patients, they become also paralyzed, and then we have some ways of checking what would help with this paralysis. Similarly, there are some fish that can be used, and the fish are vertebrates, so they are closer in a way from humans and the other models have mentioned. And they indeed have some very nice motor neurons that we can visualize. And you can see when there is a mutation that is associated with ALS, these motor neurons do degenerate. You don't see it very well here, but actually we can monitor how much the fish swim, and the fish who have ALS mutations, they don't swim as well. And so we can really follow what would happen if we test a therapeutic in these models to improve this type of behavior. So of course, fish, yeast, it's all very far away from human. It's not mammalians. And so there are many studies that are using closer models and closer animals, including non-human primates and also rodents. Of course, for obvious technical and as well as ethical reasons, most of the studies are actually done in mice, despite the fact that the mouse brain is much smaller than the human brain. But we actually know that a lot of the architecture and the function of the brain is conserved between human and mice, which really allow us to look at different things. And in particular, these motor neurons that I've told you, they are really similar between the human and the mice. And you can see in some mice where we introduce again a mutation that is associated to ALS in patients, these motor neurons in the spinal cord, they degenerate compared to the normal mice. I've told you these motor neurons, they go to the muscles and they need to connect to the muscles. And again, this connection is lost in ALS models with mutations that are introduced from the patients now in some mouse models. We have, of course, looked at survival, and in my previous slide, you could see these mice with an ALS mutation. They were dying earlier than control mice. But I think it's also very important to point out that we have very more subtle ways of analyzing this, because we are not only interested in mouse survival, but also in the function, in particular the motor function. And we can, really, these are only a few examples, we can really assess the behavior of the mice. That's just an example where the mice, they like to go to a black box, and so we put them in the center and we count how many times they many times they do mistakes before they find this black box. And so over time, they are supposed to learn. And so if you see a deficit, it really tells you they have a deficit in learning. There are things that are more, motor function. So they like to run on a wheel, or they like to hang on wires. And so we count and we measure how much they do this. And really this type of approach is, even if you have a mouse model that does not die, does not recapitulate really what happens exactly in human, but because they have some deficits, it allows you to test therapeutic approaches. And that's an example of a mouse model we had made that express a mutation in the gene for C9orf72, which is by far the most common genetic cause in both ALS and frontotemporal dementia. And we developed, in collaboration with Ionis Pharmaceuticals, some antisense oligonucleotide that rescued the phenotypes in mice. And that leads to a clinical trial that is ongoing, led by Biogen in ALS patients with this particular mutation. I have already mentioned how it's important to look at more subtle phenotypes. And these are very examples where we try to mimic in mice studies what is going to happen during the clinical trial. And so in patients, there is some what we call EMG are things to look at the function of the neurons that are very commonly done in patients. And now we know how to do that in mice. And we can really see that so this is an ALS mouse model. And then when we treat these mice with, in this case, a particular antisense oligonucleotide, it does rescue. Similarly, there are some what we call biomarkers that we can follow the disease progression in patients, and now they are used more and more in these mouse models to develop therapeutics. And this is an example of neurofilament, which is highly used now in ALS patients and other neurodegenerative disease. What the mouse studies have also told us, and I think it's extremely encouraging for patients, is that actually treatments that are efficient in stopping the expression of the abnormal gene are actually able to restore the function of the mouse and of the neurons. So it's not only that we stop the progression, but we also rescue some of the function. And that has been seen even in some very, very severe mouse models where in this mouse, they die in a very few weeks. And if we stop the disease process, then they can really restore their ability to run on a wheel and to do things. And this is because even if we don't replace all of the motor neurons that stay here are actually able to connect with more muscles and to be more functional. And so I think that's extremely encouraging for therapeutic development in ALS patients. All of this that I've said is what we have learned and advances, but we need to recognize that these are models. They have many challenges, and it's very difficult sometimes to predict how what we see in, for example, a mouse model is going to translate when we will go to patients. And one of the challenges is really what we call the reproducibility. And so this, I think everyone should be aware that, when there is an effect that is seen in mice, it's not always reproduced when the same study is being done later. And that's an example that I think illustrates quite well, where there was a mouse model dying at a certain age, and when these mice were treated, they died later. So that was reported as a beneficial effect, right? But then what was clear was that this study was done only with 11 mice for each group, okay? But when researchers looked actually at this mouse model in thousands of them, and here it's 2,000, as human, mice don't die all on the same day. So there is a variability. And if we account for this variability by looking at many mice, we realize that actually this treatment was not better than what most of the mice usually do. And so that really triggered a lot of discussion in the field and some recommendation really on the robustness of the studies and the fact that we really need to use a large number of mice to really have some strong results and to always be very careful when the effect is really modest. Another issue that we are running into is that, again, they are not humans. And I told you some of the neurons are the same, many things are conserved, but there are molecular mechanisms that are not conserved. And again, this is an example of a TDP-43 that we know now what TDP-43 does, but it does not do exactly the same between human and mouse. And there is here an important gene that is altered when TDP-43 is mislocalized in the patients. And we know that that's not happening in mice, right? And so this is where really other models, including IPS models and IPS neurons that comes from human are very valuable. And so these are methods where we take blood or skin biopsy and then we can reprogram these cells and grow them in a dish, including neurons. They have many limitations. It's in a dish, you don't have exactly what you would like. Not all the cells are there, but they are humans. So some of the mechanisms, you can really probe them. And that's an example where, I told you that if TDP-43 is abnormal in neurons, this is not good. Indeed, in this SAS, cannot regrow. And we have identified, some molecules that actually target this gene. I've mentioned STATME2 that allow them to regrow. So that was very important to do that in cells. But the issue is that when you find a compound that work in a cell culture, we know now from many studies that they don't always work if you go in vivo. And so we need models to be able to test them in vivo. And I told you for this STATME2, this is not conserved between mouse and human. And so what we had to do was actually to introduce a part of the human gene to the mouse gene to really be able to test these molecules. And that's exactly what we have done. Now we know that this molecule using a mouse model can do that. So I'm going to end here and really, I hope that I have conveyed how it's very important for us to understand the disease mechanisms. Otherwise, we cannot know where to go to develop new therapeutics. And that goes through many different ways and many different models. Of course, using the genetics, using post-mortem tissue from patients is really fundamental and has helped a lot. But then from this information, we need to develop models and to be very robust in the way we use them and recognize the challenges of using them to test therapeutics. So thank you so much for your attention. And I think Dr. Beria will take care.
Thank you. Fabulous. Oh, right. I need this and that. Okay. Thank you very much, Clotilde. I want to just highlight a couple of things in that talk that are just stories that are really astounding. In the Ataxin-2 story, there were investigators who went to yeast cells and they found that they could manipulate Ataxin-2 in yeast cells. And what they found out of those yeast cells, they could then translate into other models of the disease and then into people, and there's now a trial. So that pathway of sort of going from something as fundamental as yeast into people is real and is happening and is driving a lot of the most exciting science in people today. And the other thing that you said is that in mouse models, we see that if we can stop the disease, we can actually see recovery of function. I think that is incredibly hopeful for people who have ALS today and for some of the things that we're doing in people. I'm going to change topics a little bit here, or maybe even profoundly here, and talk about developing digital biomarkers for people with ALS and for hastening drug development in ALS. And sort of the start of this story is that when we are trying to characterize a disease or treat a disease, we need to have a way to quantify that outcome. So we need to understand how the disease is changing over time in people. And in ALS, we do this traditionally with a questionnaire. It's a 12-item questionnaire, and those items are each scored. And it asks about speech and swallowing and finger strength and core strength and walking and breathing, which are all fundamentally important things in ALS. And the total number of this scale allows us to tell, 48 is the highest, that's perfect health. And over time, that score may decline. And if we're testing a drug, we can see whether somebody declines more or less, and that's how we make a determination about whether a drug works. This has actually been a fundamental part of our drug development process for many years, even decades. And it's a good outcome measure, but it's not perfect, and we'd like to do better. One of the challenges with it is that we really do it when people are in clinic with us. And so we do it about once a month at most. And so if you imagine that we run a 6-month trial, if somebody misses one of those visits, they would really be missing almost 20% of their data. And that's a big hit to our statistics in a trial. The other thing is that we sort of imagine that the ALSFRS-R, that's our name for this, it rolls off the tongue, doesn't it? That we imagine that the ALSFRS-R declines in a sort of a linear fashion, that every month people lose 1 point, for example. But the reality is that it's much more complex than that. We can see here that there are groups of people who progress at different rates on the ALSFRS-R and in different patterns. And we don't actually know whether this sort of variability reflects differences in disease progression or differences in how our scale works. Because one of the challenges with a scale like this, a questionnaire, is that if someone's feeling happy on a particular day, their score could be higher. And if they're feeling down, their score could be lower, even if their actual underlying function is the same. And so that's something that we'd like to get rid of, and we'd like to be more quantitative in how we follow things. This is a place where we could make some improvements using digital biomarkers. Digital biomarkers are things like data that comes off of a cell phone or a wearable device, a Fitbit, an Apple Watch. These kinds of things give us digital data in a high density. They might allow us to get much more frequent data acquisition, reflect changes in health quickly and accurately, quantitatively. They could be accurate and statistically powerful, and they might facilitate more efficient clinical trials. And I'll talk about some examples of those things. And there's also a possibility that they could help us in clinic or even with the diagnostic process, something that's not easy in ALS. I'm going to talk about 2 basic categories of digital data collection. One is something that we call active data collection, active digital data collection, where participants perform a task and then we record data about that task. So imagine that we have somebody wearing a wearable sensor and they walk a certain distance, and then we use that wearable sensor data to sort of quantify how that task went. Passive data collection is when participants sort of live their life and we get data about that. So imagine an app on a cell phone that's collecting data in the background from an accelerometer. That's an example of passive data where people don't really have to interact with it much. And there are different sort of upsides and downsides to each of these types of data. So active digital data is constrained. That's a good thing. It allows us to ask and answer a precise question. What is this person's swing phase of their gait? something very specific like that. And it gives us really high quality data if we choose the right sensor. The challenge is that there can be lower compliance. So we might do this at home and have people do a task every week. But people, unlike mice that like running on wheels, people forget to do things, and they decide they're not going to do it that day. And so there are some challenges with active data collection that we don't have with passive data collection. But it allows us to get more frequent data, particularly if we do it in someone's home. So we might have people do the ALS functional rating scale on their own on a cell phone at home, and we could do it every week fairly easily. So we already are getting more data, but we might also have them do a walking task or a breathing task at home. As an example, we have an ongoing digital data study, where we have people doing active tasks. We call it the digital ALS study, and actually it's hardly ongoing anymore because yesterday was the final visit for the final 50th participant. But we gave people an iPhone and an Apple Watch that they could use for this study specifically, and we asked them to do cognitive tasks. These are puzzles that they did on the phone, or spelling tasks. We had fine motor tasks, almost like little video games where people manipulated things on the screen using their fingers, and we timed those and recorded how well they did. And then we had gait tasks where they wore a watch and the phone, and they did walk a certain distance for a certain amount of time, and we could record that. And finally, we made speech recordings of people reading a paragraph, and also asked them this ALS functional rating scale question, what we call a patient-reported outcome, or PRO. So the sort of lowest hanging fruit in what we're doing is to ask the question, look, if we have people take this ALS-FRS-R at home on their own as a questionnaire, does it give us data that's comparable to us bringing them into clinic and walking them through the paces and having an examiner actually ask them the questions and interpret that? And the answer in a couple of studies, and we've done more since this, has really been a resounding yes. They correlate very, very highly. They're not exactly the same, but boy, they correlate really, really highly. And we can get a lot more data if we ask people to do it at home, and that's a benefit. We also have moved to doing speech recordings, and these speech recordings allow us then to use machine learning and sort of big data techniques to examine these read passages for quantitative measures of how people's speech is performing. And people with ALS do have speech changes. So in the first figure here called pause analysis, what we're doing is adding up all the time that people are not speaking when they're reading a paragraph. And then we're saying, how long is the pause time during that paragraph. And what we see is over time, the pauses in reading go up as a result of disease progression. We don't see that in healthy volunteers. I'm not showing that here, but we don't see that. We also look at speaking rate. So 150 words per minute is a fairly standard speaking or reading rate. And what we did in this analysis, overall we see in people with ALS, there is a decline in speaking rate over time when they're reading a read passage. It's not necessarily detectable, and we can see these changes before a listener can detect a change in somebody's speech when we're using these machine learning algorithms. But we also did something here which was to say at the outset of the study, are people speaking normally at a normal rate, or are they speaking at a slower than normal rate? And what we find is that if people are speaking at a slower than normal rate, they have a decline over time in how quickly they speak. If they're speaking at a normal rate at the outset of the study, their speech might decline and it might not. Now, this reflects something that we understand about ALS, which is that it often starts in one part of the body and spreads to other parts, but the timing of that spread is unpredictable. So we might see someone who develops first foot weakness, and then weakness in the leg, and then weakness in the arm, and then weakness in, that affects speech. But we don't know exactly what the time course will be for them to develop that weakness. This would provide us a way to say, look, at the outset of the study, these are the people who have a slow speaking rate. Let's focus our analysis on them, and it can give us really benefits in designing a trial and being able to run a trial on the people who are being affected in the region that we're measuring. So passive digital data is slightly different. It is really good because it's real world data. We can measure how people live their lives. And if we know how that disease is affecting their life, and if we're testing a new drug, we can ask the question, is our drug affecting how people live their life? There's a high compliance typically because we're not asking people to do anything differently. And we can use true big data techniques on this because we get millions of data points rather than dozens of data points. The downside is that there's also a lot of noise in the data, and we have to have algorithms to understand what to do with that. So just to give you an example, we've looked at measuring something monthly, we've looked at measuring something weekly, but with passive data collection, we can literally measure things minute by minute, second by second, 24/7. And that is, a giant data set. One of the challenges with passive data is that we not only need to collect the data, but we need to be able to analyze it in a meaningful way. So this is what GPS data looks like for a number of participants before we put it through an algorithm to understand that data. It really wouldn't tell us anything. We couldn't make any sense out of that. But if we apply an algorithm, we can now say, this is how far people are traveling each day. And if our algorithm is doing a good job, it will allow us to see, as we do here, that people travel less distance over time. And this is from a cohort of people who have ALS. This was a program that was on their cell phone. And it simply says that people over time traveled less distance. So there's this important step of sort of being able to analyze the data and so it will show us the truth. That is not something that we can overlook, and it's not something to be taken lightly about passive data. But there is a really powerful sort of central tenet to getting passive data. And there's also a real opportunity because cell phones are loaded with sensors, accelerometers, gyroscopes, GPS, logs of calls and logs of text, not what's said, not what's written, but just when was a text received, when was a call received, when was a call made. That could be important data because we know, for example, that ALS affects people's ability to communicate. If somebody's having difficulty speaking, they may need to move to texting in order to stay connected to people around them. And to be able to understand how a disease is affecting somebody's life is a really powerful thing. And we might be able to use that data to get insights into that. And the other thing is that smartphones go with us. So every hour of the day, 91% of people have their cell phone within 2 feet of them. And I think that definitely rings true. Not everybody, there are some people who don't live this way. And we have to take that into account when we do these kind of studies. But this really does ring true. There's nothing in that statement, I think, as I look around the room and as I sort of interrogate my own feelings, I think that feels right. Sort of leaving your cell phone at home means you're going to turn around and go back and get your cell phone. Wearing 2 different shoes, maybe I'll make it through the day. GPS data. So here in that dark blue color is so this is a graph of people who are in a study. This is real data from study. I just pulled it just a couple of days ago, and you can see that it's sort of over time. So the earliest person to enter the study is up at the top, and they entered the study, and we have a lot of data on them. And then down near the bottom are people who entered the study later. The dark blue data is when we have GPS data from them. The black data is when we should have data from them, but we don't. And so you can see that even though it's passive, and even though they don't really have to do anything for us to get that data, there are times where we're not getting the data. Nothing is perfect. There are technical glitches. Somebody's GPS goes off, they get a new phone, they need to reinstall the program on their new phone. There are lots of reasons that we miss some data. Still, having said that, we get a huge amount of data from this kind of study design. And as we say, the proof is in the pudding. We can use that GPS data, and this is also sort of hot off the presses. It has never been published. We just pulled this out of an analysis a couple of weeks ago to look at the average amount of time that people spend at home. So we can sort of look at people's GPS data that we get and say, well, they spend most of their time in this location. We're going to call that home. And then we look at how much time they spend at home. And over time, over a period of about 6 months, we can tell that people spend more time at home when they have this disease. It is impacting the way they live their life. We can quantify that. And if we're developing a drug, we could give half the people the drug and half the people placebo and see what happens to the amount of time that they spend at home. And we would be able to see whether not only is the drug working biologically, but what is the impact of that drug on their life. Importantly, we also want to correlate the outcome measure that we know works with the new one that we're developing. So changes on the ALS-FRSR do correlate with the time that people spend at home using their GPS monitor as well, or their GPS data as well. In other words, when people are functioning better, they spend less time at home. When they're functioning worse on the scale, they spend they spend more time at home when they're more impaired. And that's what we want to see. It's not a perfect correlation, but they're measuring slightly different things. And another way that we can kind of decorate the data, if you will, or enhance our understanding of the data that we're getting. This is an ALS functional rating scale score. You can see, let's see. You can see over in purple on this side. These are the people who have a high score on their ALS-FRSR. They're functioning quite well. Here are the lower scores on the ALS-FRSR. And what we see is over time, or pardon me, not over time, but as we compare to the number, it actually takes people longer to complete this survey. So if someone has a low score, it takes them longer than people who have a high score and are functioning better. That's both a good logic check and may give us a new variable that we can incorporate into our statistics and have a sort of a more powerful analysis. So there are some simple things like this when we move from a paper and pencil task to a digital task that give us new insights into the disease. I could go on. We have a lot of data like this that we're exploring. Some of it, I should say, does not work out. There are things that we try to analyze that just don't seem to give us the data that we would expect. It's complicated. Looking at gait, for example, looking at how people walk in ALS is complicated because at some point they start using a cane and that changes their walking again. And so there's a lot of factors here, but there's also a lot of data. And so we're working through some of those things. But I want to turn my attention to another project, which is perhaps even more exploratory. And that is the idea that maybe we can use internet search data to identify people who have ALS. And the idea for this really came from the fact that over time, I've seen people come in to get diagnosis of ALS, and fewer and fewer people were surprised by this because more and more people said, well, I've been reading on the internet, which means that they've been searching on the internet. And one of the problems we have in ALS is that it takes over a year to make a diagnosis of ALS after people experience their first symptoms. That's a really long time. It's a very stressful process. And it's challenging to know how to circumvent that. And a lot of things that we've done have not really worked to decrease that time from symptom onset to diagnosis. But there is a lot of data that we haven't explored, and that data is on people's search engines. And so, the thought occurred to me that maybe we could use this. And it's a fraught idea. There are a lot of reasons that may or may not be a good idea. And it's also possible that there's just noise there. But I met two people who were sort of interested in this idea as well, Ernest Frankel, who's at MIT, and Elad Yom-Tov, who's at Microsoft Research. And we began to explore this, and Elad has actually looked at how search engines can track real life events in a book that he wrote. And he looked at people who were women who were pregnant and what their search terms were over time. And you can actually track through time, through a pregnancy, from sort of pregnancy tests to heartbeat to baby moving all the way up to into labor and my newborn. So that in real time, search engines can give us, at least theoretically, insight into what's going on in someone's life. And certainly ALS is impacting people's lives. And so we had this idea that we could do a two-part study. The first part was to use search engine data to identify ALS as well as we could. And what we did is we essentially said, we're going to make a group of people who say to their search engine, I have been diagnosed with ALS. What now? And it wasn't quite that simple. We had a lot of terms, and we were pretty rigorous about this. And out of 100,000 search records, we picked out something like 300 that seemed like they may really have ALS based on this. Now, we can't verify that. We're not sure of that, but it seems like there's a reasonable likelihood. And then we compared the data between those 300 and about 3,000 other people who didn't seem to have ALS. And I'll show you that data in just a second. The second part to this study will then be to go back to people to do what we call a prospective study. That is to say, we're going to have people who have ALS register for this study and anonymously sort of contribute their search engine data so that we can look at that compared to healthy controls and say, do we replicate this? Are we really sure that this is right? But in the first part of this, and so that's ongoing now, actually, people are signing up on the web for that. In the first part of this, we found that we could actually differentiate people who have ALS from people who don't have ALS based on their search results. And there were a number of factors in this. One is that autocorrect is triggered much more frequently in people who have ALS than people who don't have ALS. And the other is that the ALS group searched for rare terms more. Now, I want to be very clear. We're not searching through for exactly what people are searching for. There's a machine learning algorithm that spits out these figures. But there are well-defined rare search terms and more common search terms. So that you understand the sort of detail at which we're looking at this data. And the rare queries are much more commonly used in people with ALS. There's a lot to understand there. Why is that the case? Not 100% sure. Is it related just to ALS? Is it some other, something else going on cognitively? That there's a lot of things that we don't understand, but these were some of the findings. The very, very early hint into the prospective study is that we have a handful of people who've already entered into the study, and we're finding exactly the same thing in the people that we know have ALS. So really sort of tantalizing. The next question that we want to ask is, how early can we detect these differences? Is it after they came to diagnosis, or is it before they came to diagnosis? And if we can do it before they came to diagnosis, then there could be real benefit here in some way or another. There are a lot of dystopian ways you could take this. That's not the intention at all. I think right now we're just exploring the idea. The idea behind these digital biomarkers is that we can get more data than what we're traditionally getting. We can do that at a lower burden. We can allow more people to participate in research. We can make our clinical trials easier. We can make our clinical trials more efficient. And that's really important. And this is the end of what I have to say. A huge number of people go into sort of getting this data. Huge thanks to all the patients who participated in this. But with that idea in mind, that we can really change the way we collect data and how we apply that to clinical trials, I want to introduce Sabrina Paganoni to talk about how trial design is changing in ALS, so that we're sort of revamping the whole structure of how we're doing ALS trials to bring efficiency and to find effective therapies, which Sabrina Paganoni has been really a leader in the field for. So Sabrina, please take the...
Okay, wonderful, thank you. So it's really a pleasure to be here. I've been here before in past editions, and today you already heard about great science, and both in the lab and in clinical research. And today I sort of want to show you the end result of this, what happens when we have great science in the lab and great clinical research. Ultimately, the reason we're doing all of this is to develop new treatments for people with ALS. And so I've been fortunate to be involved with several trials in collaboration with Dr. Beria as well, and other colleagues who, like me, see people with ALS in the clinic. Like Dr. Beria, we work in the same ALS clinic. We are physician scientists. So we have really this unique opportunity to meet with people with ALS on a daily basis and interact with them, and at the same time, work on designing and conducting clinical trials so that we can bring these innovative products, new drugs that are developed in the lab, new tools that are developed, such as digital biomarkers, to clinical trials. So today I'm going to spend about 5-10 minutes telling you more about some emerging treatments and some innovative clinical trials, which in the end, we hope will bring new treatments for people with ALS. And the reason I'm very excited to be here today is because 2022, I think, will be remembered as an important year for ALS. And the bottom line is that there are 3 topics that I think are really gonna change, and kind of future books written about ALS will include 2022 as the year when we had maybe, hopefully, a couple of new treatments approved, and I'll tell you more about those, as well as really finally the establishment, the development of innovative clinical trials so that we can test more drugs and therefore hopefully bring even more drugs to market. And finally, this is a time to create more access and more options for our patients, because that's what they want to do. What we hear all the time from our patients is that they want to have more opportunities to participate in clinical trials, more options, different types of trials. Perhaps not everyone is a good candidate for a formal clinical trial, but perhaps there are other ways to expand access and give people more opportunities. So I'm going to kind of briefly summarize my thoughts on these 3 key topics, and then I know we have a Q&A. So again, I'm going to keep this brief so that we can then maybe talk more about it in the Q&A section. And specifically in terms of the first topic, I told you there's a couple of drugs that I think might be approved in 2022. We don't know yet. Obviously, until we get there, we won't know. But 2 of them are, the first one is actually an oral form of edaravone or Radicava. You may be familiar with one of the 2 drugs that's already approved for ALS. It's an intravenous form of edaravone or Radicava. And I hope that the FDA will approve the oral form. So that means making it much easier for people to access the drug. It's the same drug, but certainly much easier to take. So, and we expect the decision by May, at least as announced by the company. And then the second drug that I want to talk about is called AMX35. It's been developed by Amylyx Pharmaceuticals. And we really work closely with them as an academic industry collaboration to help design and conduct the trial. And so I'm going to talk a little bit about that. Then I will talk about a trial that has not produced, final results yet for all the investigational products, called the Healey ALS platform trial. And that's important because as new drugs are approved, again, maybe we get 2 new drugs in 2022. It's also important to continue to develop the pipeline and test as many drugs as possible in the pipeline, because obviously as we bring new drugs to market, we want to continue to look for more cures. And that's where the innovative trials like the Healey ALS platform trial come in, with the goal of making things faster and more efficient. And lastly, I'm going to talk about something that I think, I can speak for many of my colleagues and myself. It's really a pleasure to continue to work with the patient community. I think we have found new ways, even during the pandemic, to interact with patients and have a continuous dialogue. So I'm going to talk a little bit about that. Starting with the Amylyx drug, AMX35. So that was tested in a trial called Centaur. And both Dr. Beria and I worked on it. And we specifically, the company was founded by Justin and Josh, who are in the top picture. And they came at the time, they had the idea of the compound. They had a compound, but they didn't have the know-how at the time. That was about 2015. To develop the drug. And so they came to MGH and they started to work with Dr. Tanzi on the left and Dr. Sukovic on the right, who are Alzheimer's disease and ALS experts respectively. And so together they worked on sort of designing the trial. And that's when I also got involved. Dr. Sukovic actually had the idea to test the drug for ALS as well. And so around 2015, 2016, we started to work together, and as well as with other collaborators, to design the trial, test an efficient trial that could provide, answers, as quickly as possible. And that's how, this Centaur trial was started. And since then, Amylyx grew exponentially, and now it's a bigger company, and so they have their own expertise. But again, we partner at the beginning as an academic group, to help them launch the trial. The drug itself is called AMX35. It's a combination of 2 drugs that were already known before, called PB and TERSO. They tackle different cell mechanisms, and that's important because we still don't know really what causes ALS. There may be more than one factor at play, but they all converge on some key cellular mechanisms. And so perhaps, just like we have seen in other complex diseases like cancer or HIV, when you have complex diseases with many different mechanisms, the first treatments that were developed for those diseases are cocktails of drugs that tackle different mechanisms until you can really get targeted treatments. So for now, we are still in this stage with ALS. And so again, this drug is basically a cocktail of 2 drugs that can also be combined with Riluzole and Radicava, again, to add to standard of care. So the beginning of the trial was really a collaboration between the academic community and Amylyx Pharmaceuticals. We also support from groups such as the ALS Association, and it was conducted at 25 sites of the NILS Consortium, which is the academic consortium of ALS clinical trial sites. And the results are summarized here. So the drug didn't stop and didn't reverse the disease. That, we want to be very clear. This is not the cure, the ultimate cure for the disease, but certainly it provided very substantial benefit, as demonstrated by the primary analysis. Specifically, there was retention of function, which is shown on the left panel, where there was a difference between people who received the drug and people who received placebo, and the difference was measured on something called the ALS functional rating scale, where there was a difference in favor of active drug, meaning the people who received active drug retained physical functions longer. And also when people were followed long-term, there was a statistically significant and I believe clinically meaningful advantage for the people who were started on active drug on the order of about 6 months. So again, people did progress, the disease didn't stop. At the same time, there was a significant survival advantage. So they lived on average 6 months longer, which obviously, it's a significant amount of time. So what I want, and so basically to conclude this, basically the AMX35 drug story is not over yet. We are expecting an advisory, a public advisory committee at the end of the month on March 30th that will provide some guidance in terms of, to the FDA, and then the FDA will make the final recommendation by June. So again, if you think about it, over the next couple of months, we're going to have 2 different answers from the FDA in terms of oral edaravone in May and AMX35 in June. So it's possible that people like Dr. Beria and myself, when we see patients in clinic in July or so over the summer, we will be able to prescribe 2 new drugs, which obviously for us is exciting, because if you think about it, it took almost 30 years to develop the first 2 drugs. Just to have perhaps 2 new prescriptions available in 1 year, it's pretty remarkable. Now, obviously, let's say that this happens, we all hope that this is going to happen. So where do we go from there? So we need to be ready to continue to add to the standard of care. So if the new standard of care is going to be, these new drugs, how can we do even better? And that's where the Healey ALS platform trial comes in. So this is a trial that, many of us, also the previous speakers, helped work on in many different ways. And so it's really an amazing collaboration between so many, not only in academia, but also industry, patient foundations, etc. And the reason there are so many logos on this slide is because it's essentially pretty much everyone in the ALS community really supports the trial. And we have had a lot of conversations, again, with so many people who provided input into the design of the trial, and also the FDA itself has been heavily involved and supportive. So I want to explain what the trial is. And so to do that, I'm going to ask our technicians to help us show a short clip from a video that explains what's different about the platform trial.
The fight against ALS, the first ever ALS platform trial, designed to accelerate the development of effective and breakthrough treatments for people with ALS. So what's different about the Healey ALS platform trial? Instead of testing just one drug, the Healey ALS platform trial evaluates multiple treatments at the same time. This means more opportunities to find groundbreaking therapies in a faster time frame. Imagine having to build a new dock every time a different ship came to port with new treatment cargo. This is how traditional trials work. In platform trials, there is one dock to receive all the cargo and connect the treatments to their final destinations.
So essentially what we have done over the last couple of years is to work with a large number of statisticians and experts in different domains of ALS science to build this optimized infrastructure. So that now we have an optimized infrastructure to test as many treatments as possible. And so the way this works is that companies can submit their application for inclusion into the trial. And in fact, we continue to partner with people and, and continue to ask for, interest from companies. The infrastructure is now up and running. In fact, as of 2022, we have been including already 5 treatments, 5 possible, investigational products. They are named on the slide. Every drug is tested in its own regimen. That's what we call a regimen. So it goes by letter A, B, C, D, E. At this time, so far, we have included 5. And we we started enrolling in 2022. We expect the final results of the first 4 we started enrolling in 2020, but with the pandemic, that time is completely a blur. But we started exactly when the pandemic started, and now we are about to get the results from the first 4 drugs over the summer in 2022, and we just started with drug number 5. We just started enrolling, but now we're already working on adding a couple of drugs later this year, and then we continue to interact with partners for potential inclusion in the future. So again, we opened, a couple of years ago, and we expect to continue to test more drugs. So the idea is that if we already have an ongoing infrastructure that's going on, and always open, we can always test more drugs. One thing that just happened a couple of weeks ago is that actually we do interim analysis. So we continue to analyze the data in an ongoing fashion. And we figured out that the first drug, regimen A, the very first drug that was entered into the platform, didn't work. And so we stopped it early. And now that's obviously disappointing because, we wish we had positive results. But that also kind of, it's almost like the proof of why this is so important that we do trials this way, because it's, we need to get an answer, whether it's positive or negative, but importantly, we need to get an answer as quickly as possible. And so because the drug was included in a platform that was already ongoing with these frequent looks, we were able to stop it early and so refocus all the resources on the other ones and create more spots for new drugs. So that's the idea, to basically make all of this more efficient. Now, we still have a problem in ALS that traditionally only about 10% of people with ALS have been able to participate in trials despite a lot of interest. And that's because until a few years ago, there was only one trial here, one trial there. There were not many, and they were always, sort of limited in terms of access opportunities. And so we said, okay, how do we change that? So one way that we have done, so, we're doing this in multiple ways, and there's many of us who are working together on trying to find solutions. One way is to open as many sites as possible so that the trials can get close to where people live, because obviously it's hard to travel to participate in trials. And so of the NILS consortium, which is this US-based large clinical consortium of sites, a number of sites have entered the platform trial. Initially, we had a group of 52, and then 22 more were selected. And so essentially, they're being all activated and part of the network, so that if they're always up and running, we basically remove the administrative burden of having to select a site and adding another one, etc. So all the drugs that I mentioned earlier that are part of the platform trial are then available at all these sites. So it's almost like, it equalizes the opportunities across the country, because again, patients, no matter where they are, as long as they can get to one of these sites, they'll be able to access at least the platform trial. Then another thing that we've been doing is to, in the pandemic, we started doing webinars all the time. So we are online pretty much on a weekly basis, if not more, for live Q&A and updates with the community. And so this provides a forum for us to really be able to be available for patients and be able to answer their questions on an ongoing basis, which I think is really a change compared to the way before, there was access, I guess, to researchers, et cetera. And so we continue to invite a lot of guest speakers to be able to be available, on our webinars. We also created this patient navigator, which is a central resource for people to contact us, if they have any questions. And this is in part funded by the Tambourine Collaborative, which is a great group that also helps us think about ways to increase access to trials. And we do these weekly webinars, we have newsletters. Again, we really have put sort of access and inclusion at the forefront of our efforts, so that again, we can not only run trials, but run them in a way that are accessible to patients, no matter where they are, no matter their background. So we've been trying to do this as a team. I really appreciate, the opportunity to be here and be able to interact with all of you. And of course, we have a great team. I think we could have many slides to thank everyone and all the names. I just give a collective thanks to the previous speakers and our collaborators and all of you for being here. And again, we'll keep providing updates as we go along. Thank you.
I was wondering if there has been much attention to how to identify the potential or the actual conversion to ALS with people who have frontotemporal dementia.
So I'm going to repeat the questions for people who are listening or here. And I want to make sure I get the question right. Have we put thought into how to identify the development of ALS in people who already have frontotemporal dementia? Yeah. So it's a great question, and I want to just back up a little bit to just explain that frontotemporal dementia and ALS are diseases that sometimes are linked genetically, sometimes are linked by pathology or disease mechanism, and we do see these co-occurring in some people. Frontotemporal dementia is marked less by memory loss and more by changes in attention and personality. ALS is a disease of neurons causing primarily. We know now that there's a lot more overlap from these before. That if we look deeply, there are mild motor deficits in many people with frontotemporal dementia, mild cognitive changes in many people with ALS. When we focus on kind of the more fundamental disease, less symptoms. This is complicated. So there's actually a fair amount. I'll start, and then I may pass out to you to sort of put to get some of the basic science or animal model kind of understanding of the connection between these two. And Sabrina, if you could give some further impressions on the clinical. I'll just say from the sort of outcomes perspective, it is sometimes difficult to do the really deep kind of cognitive testing in people who have motor deficits, and sometimes difficult to do the really detailed motor testing in people who have cognitive deficits. And so I think we miss this a lot. I also think that because of the way we're trained, either in sort of neuromuscular medicine, focusing on strength and sensation, and that's where a lot of the ALS training comes from, or in cognitive and behavioral medicine, focusing on sort of cognition and thinking. And it took us a longer time than it should have to probably understand the overlap of these. So there are a lot of challenges, but there's a lot of work going on to try to understand when people with ALS develop cognitive change, if they do, and when people with with cognitive change or FTD develop motor symptoms. There's some data that says when, I can think in ALS anyway, when people with ALS develop cognitive change, it tends to happen fairly early in the course if it's going to become fulminant, dementia, frontotemporal dementia. I think that's still early and needs to be followed up. I am not actually as aware that data exists on the other side, that if people with FTD are going to develop motor change, that happens early. But that's a really interesting question. Clotilde, do you want to talk about the overlap of these two from the model perspective?
Yes, I mean, I think a major thing is that before it was two different fields, right? Researchers working on ALS, researchers working on FTD, and with the recognition in patients that there was this overlap, we are learning a lot from each other. We are sharing tools. We are right. So I think it comes back to what you were saying about training, right? You are trained in something in looking at motor neurons, but never thinking about cognitive changes. For example, if you're an ALS researcher, and this has changed a lot, as you can imagine now that we are modeling the models I've described, like in flies or even in mice, maybe it's a bit different, but also the more simpler organism, we cannot imply whether it is ALS or FTD, right? These are the same models where we put mutations that can lead to either ALS and FTD. I think as a researcher, I see myself now on ALS, FTD. I don't do the difference because I think in terms of mechanism, we really don't have, reasons to dive. Where I think there's a clear lack of understanding is why some patients with the same mutations or in the same family are going to develop one or another. There are some interesting studies on is there another gene that maybe is going to favor either one or either the other ones, and I think that this is the most promising results come from there. But it could also be something more environmental that, let's say a traumatism, or like, I don't want to go into what other risks are possible, but where you could imagine that depending on where it starts in patients, it might develop towards this. I think there's a lot to do in terms of understanding this overlap. It's relatively recent that we have recognized it. I think really, 10, 15 years that
Yeah, I'll just add, you explained the clinical situation very well. I'll just add that from a clinical trial perspective, the two fields, ALS and FTD, have been traditionally very separate. Again, it's just a matter of training. Like, I work on one topic and you work on a different topic. And more recently, we started to interact a little bit more. So, for example, I've attended some FTD conferences where they actually asked me to talk about ALS. And vice versa. So, initially, like 5 years ago, I was like, okay, why do you invite me? But then when I went there, I started to recognize the importance of that. I would say there's still a lot of work to do because when you have this overlap, it can get tricky, to study, to do the clinical trials and really, get the best outcome. We still have to figure out the best outcomes and what to measure really when there is overlap. But certainly the conversation has started.
But never letting the idea of building a bridge and walking over at the same time dissuade us. We actually are already, I think you heard from Sabrina about this novel platform trial design. We're also already beginning to see what we call basket trials, where we use the same drug to sort of see if it has an effect on two or more different conditions. So we're seeing some basket trials now. We're running one and designing another, looking at sort of the effect of a single drug on people who have FTD and others who have ALS. So we're really trying to bring those together. But there's real challenges in how we design that. So it's an exciting time.
What are some of the hopeful things that we can look forward to in the field of ALS?
That's a great question. Sabrina, do you want to start?
Sure. I think that, if you look at the pace of research and development over the last few years, I would say that the discoveries have been exponential. And I know it's not a very satisfying answer, but I would say that with the recent discovery, if you really look at the curve of the discoveries, it's really astonishing. So, for example, maybe you can tell us more about it, but the discovery of new genes and new mechanisms. We now are really building the pipeline of drugs. That doesn't mean that we're going to have drugs today or tomorrow, although we are hopefully having a couple more this year. But I think that really you need the pipeline to grow in order to be able to have results. And I think that this has happened over the last 5 years or so. And now that we are also thinking about sort of how to test all these drugs, for example, the platform trial, but there's other innovative trials around as well, I think we're going to see an acceleration of positive results.
I think related to that is that most of these pipelines that you are talking about, it's much more targeted towards specific mechanism. I think maybe before we are thinking, or maybe that could help. Now we have a better view of why it would help actually to do that on the neurons, or to do that other thing. And we have also a better view of these different subgroups or ways that the neurons can be killed, and that might not be the same in all patients. And so we are really trying now to get to this more specific targeting therapeutic development, maybe for subgroups of patients, but that has more chance to be efficient because we know why.
Right. Yeah. I want to take one step back on something that we didn't touch on it in so much detail, is that something like 85% of ALS happens sporadically, and about 15% happens from a genetic cause that's carried in families. And that genetic cause is not always the same, and there are multiple different genes. And so those genetic causes maybe give us a foothold into understanding the first changes that could set off ALS, and also because of new tools that we have, give us maybe our first crack at really targeting a very specific molecule or DNA or RNA that's sort of kicking off the disease and driving it. And we're seeing those trials sort of in the clinic now, and I think we'll see an expansion of them. And Clotilde's work is using a lot of these tools to target these gene mutations in models, and that's what's spawning all of these trials. And we have new ways of testing these drugs in clinic, and Sabrina talked about that. I think this is really important. But the other thing to recognize about this is that as we discover genetic modifiers of disease, how quickly does the disease progress, who's at risk for the disease, we can move, I think, out of that 15% of familial or inherited ALS to the sporadic disease. And Clotilde mentioned one of her really just fundamental contributions in recent years. There's so many, but it's a molecule called Stathmin-2, which may be a target for genetic therapy and in the sporadic population. So I think these tools are really driving us to a new era in ALS. I think that's incredibly hopeful. And at the same time, we're seeing drugs that are really built on powerful observation in animal models and in patients come to fruition, like the Amylyx drug that Sabrina talked about, which is not specific to any genetic form of the disease or any sort of specific biology in a subset, but seems to apply to really everybody that we studied in the trial. So I just think it's remarkable. One other point on this, which is maybe a little bit out there, but how much progress we can make in science is determined by the tools that we're using. And so if you think about sort of cooking in the kitchen, cooking went along, it was cooking, and then the microwave oven became a tool. And it really changed the way that we could cook, and we could reheat things, and we could, do part of what we're doing cooking. And there were some missteps, there were a few TV dinner missteps along the way, and I've had some, mashed potatoes that did not come out well from the microwave oven, but on the whole, really, really changed the way that we could do things in the kitchen. And, we see this with the personal computer. The personal computer came out. What was the effect of it? I mean, you can't even begin to say what the effect was, but there are people making spreadsheets and drawing designs and, fundamentally changed the way that so much of what we do happens in the world. And we're seeing those changes in medicine and in the lab and in ALS. Not from the microwave.
I'm fascinated by the choice of the microwave.
Well, you do things faster. But from techniques like, and Clotilde, I'd love to ask you a little bit. We hear a lot in the press about CRISPR and sort of this is one of those tools. This is the sort of like the microwave oven. You can speed things up to cook them faster. How are you seeing that change what you do, and what do you sort of see for that? And maybe just a brief explanation of what CRISPR is.
It's a great question. I mean, it's maybe you have heard in my presentation, I was always saying, we introduce this mutation and we to make models or we modify this part of the gene, and that is actually not easy to do, even in a cultured cells, even less more complicated in an organism, and so. We could do it for decades, but very inefficiently. And I think what CRISPR is doing, it's just a tool, like a new microwave, that increase exponentially our ability to make these changes. And so basically we are able now, each time that a new gene comes, is discovered in patients to modify cells in culture, or even to create new animal models who are going to express this particular mutation at a speed and at an efficiency that was incomparable. So that is more in terms of as a tool for research, and it has changed research in all domains, I think, definitely. And now the question is, can it be used as a therapeutic tool? And we are definitely not yet there. There are many challenges, but it's true that this ability of maybe removing an abnormality that leads to disease in the genome. It's really going to the source, right? And so there are a lot of efforts on testing the efficacy, but of course very much a safety, and it's very well, it's very well thought of and restricted, of course, because of all the ethical risks that are around genetic manipulation. I don't think CRISPR's changed these risks. They were existing before. There is a lot of debate and discussion in the research community. It's just that these tools allow us to do it more efficiently.
Can you guys speak a little bit to how do you see your work and your findings translating or making a positive impact to other neurodegenerative diseases?
I can already start because when you were so we have talked about ALS and FTD, and that has been an obvious change. But I've talked a lot about TDP43, this protein that is abnormal in all ALS patients. Actually, it was recognized later that it's also abnormal in almost 50% of the patients with Alzheimer's disease. So this connection was not done before, but all I've talked about Ataxin-2. We have talked a little bit about Statin-2. They sound the same, but they're very different. But these are approaches that are towards TDP43 abnormality, and now we know that might actually impact different neurodegenerative diseases. So again, recognizing mechanisms and seeing what are the common mechanisms is really a way for us to make connections and to see how we can develop therapeutics that could benefit a larger population.
Sabrina, do you want to talk at all about how DIAN-2 sort of connected with the platform
that's
trial?
great. Yes, I mean, you're right. That's another great example. So I spoke a little bit about the idea of the platform trial and testing multiple drugs at the same time. And really, when we had this, we got this idea from cancer, to be quite honest, and that because they've been using that type of approach in oncology for a number of years. And then when we wanted to bring it to neurology, we then spent, you and I and others spent one day at WashU because they had already done a platform trial for Alzheimer's disease. And so again, that speaks to the idea of we really learn from each other, and sometimes it's actually really refreshing to bring new ideas from other fields. I do want to say that a great mentor of mine, actually I have a great mentor who told me once, don't go anywhere without having a scientific magazine in your purse, because otherwise, when you have free time, you can read a paper. And I had to say, I've been doing that. So that was my first mentor. Then another mentor told me, you should always bring a scientific paper of a topic that's not your field. And then I started doing that, and I realized, wow, that really brought me to a different level, right? Because it's not just reading about your own field, but it's really reading about what others do that really helps you bring new ideas into your own field. And so I think it's this wonderful collaboration with other fields that really helps us bring new ideas into ALS.
And I'll just expand on that with one specific example, which is, I work a lot on biomarkers. I talked a little bit about what we call digital biomarkers, but I also work on biomarkers in blood or spinal fluid. And a project that we ran a couple of years ago was in the gut microbiome. So doing essentially a stool analysis to look at the bacterial makeup of the gut. And we found differences in people with ALS and then controls. And we also connected with people who are doing this in Alzheimer's and in multiple sclerosis, and now have a much bigger project looking over time, comparing across neurodegenerative diseases, and looking at how the gut microbiome impacts inflammation in the body and how that might be related to some of these diseases. So that's just a specific example to sort of illustrate, what Sabrina brought up, that they're really, in lots of ways, we're finding sort of ways to compare and bring findings from one field to another.
I was just wondering, why is the time frame between symptoms and diagnosis, why does that seem so long?
Yeah.
Like you mentioned, a year?
Yeah. Yeah, it's about a year. Maybe sometimes when we look, it's a little less. Sometimes it's a little more, but that's about right. You know, it's a complicated question. I think one of the reasons that it can be a long time is that ALS is an uncommon disease. I bristle a little when we call it rare. There are some good things about it being a rare disease when we think about sort of regulatory pathways, but the reality is that the risk is 1 in 300 or 1 in 350 lifetime risk. So it's an uncommon disease, but it's uncommon enough that it's not going to be the first thing on anybody, probably appropriately so, if somebody came in with a very slight complaint. And so we need to work through what that means. But on the other hand, I also think because it's uncommon, it doesn't come up quickly to the top of the list. And then another reason I think it doesn't come to the top of the list quickly, and I think this is changing, is that we have not traditionally had a lot of directed therapies for it. But as we get more, there are 2 in front of the FDA right now this year. As we have more of these therapies that really can change the course of the disease, particularly when they're implemented early, I think we need to work on ways to change that. And there are some efforts to do that.
Yeah, no, I completely agree. I mean, I think in the end, there are a few factors that we can modify and some that we will never be able to modify. In other words, because the onset of the disease oftentimes is subtle and takes some time to fully develop, and some of the symptoms are, can be common to many other common diseases. It's always gonna take some time. It's not like let's say an injury or an orthopedic injury that happens on a certain day and when that started, right? So that it's always gonna take some time. That said, there are many things that we can do to try to improve the system. And there's many of us who are working on sort of different ways of improving the healthcare system, raising awareness. There are multiple efforts going on in this way. Leveraging electronic medical records can, just like you mentioned, sort of searching, observing behaviors of people searching the internet. Can we actually search medical records to try to retrieve, red flags? And alert physicians about possibilities of potential diagnosis. So I think hopefully over time we'll be able to reduce that delay. I don't think we will be able to completely, make it, super fast diagnosis again because of that reason. I do love your point about the fact that as we develop more treatments, the urgency to really diagnose early is going to continue to increase, and that's why I think there's also many companies that are now looking into that, because really the sooner you intervene, the better.
Now I'm going to pick that back up because I want to think a little bit further ahead, which is to say, we inherent to what Sabrina and I talked about is that we don't have a single test right now that is sort of specific for ALS and will sort of make the diagnosis. We have to rule out a number of diagnoses first. We're trying to tackle that problem in a sort of a very focused way. We have an initiative at Mass General that we call the Initiative to Prevent ALS. And at the centerpiece of that is a study that we call the Dominantly Inherited ALS Study, or DIALS study. And this is studying people who come from a family where a genetic form of ALS exists and who might be at risk of developing the disease. And we work with them to decide whether they want to do genetic testing for themselves. But whether or not they would like to know those results, we can collect blood and spinal fluid and clinical information, and we can really begin to launch an effort to find a biomarker that that might herald the onset of the disease. We already have a biomarker called neurofilament, which is not specific for ALS, but does seem to rise either at or before the onset of symptoms. And so in people with certain genetic mutations, we can actually monitor for neurofilament rises, and even before symptoms happen, we have a trial right now where before symptoms happen, we can launch treatment with an experimental therapy if neurofilament's going up. So that's sort of the first crack at this, but I think we can get better and better when we have the resources from these familial forms. All right. Thank you very much for attending today. This has been great. Thank you.
Thank you so much.